All right. So now we're going to start another very important. So morning, what we started is the unit 13. And now we are starting another very, very important unit. That is the unit 5. You know, I encourage you, even though you are from a botany background, even though you're from microbiology or biotechnology, that you have never read the concept of developmental biology. But I sincerely request every one of you compulsory read the concept of the developmental biology without leaving it. The major reason is that this is one of the major unit where you can't get bias in the marks. I can tell you very clearly, suppose example, it is a JL or DL exam. Undoubtedly, you can get 7 to 8 questions. Even in the CSIR as well as the set level, we get minimum 6 to 7 questions compulsory from the unit 5. One of the good scoring unit, and one of the unit, every single time the questions will be repeated more oftenly. All right. So now let's start to read this unit little bit elaboratively. Okay. I don't teach faster. At the same time, I don't teach very slow also. I want you to understand it correctly. Now, what is this concept called as developmental biology? Okay. Why they have given the name developmental biology actually? Why they have given the name developmental biology? Okay. Now listen. What exactly the meaning of development? How can you give the definition of a development? Development means simplest beginnings throughout the, through the final. A simple example, friends, we all people know very well. Initially, what happened? Sperm and egg fuses. It is going to give rise to the zygote. Now, you know very well, the zygote undergoes rapid divisions and it is going to give rise to the blastula. And blastula is going to give rise to the gastrula. Gastrula giving rise to organs, which we can call organogenesis. Then organs are basically forming organ system. All the organ system come close together. They are forming the body. So how can I say here? We have actually started with, gametes, that is a sperm and egg. So we just started with the simple cells and finally we have developed into a complex body. That's the reason we can say that development is a process which contain simplest beginnings through the final. That is what generally we can call development. Now, every student try to understand me. What are the major key events in the development? This is what exactly we are going to read in the chapter of the developmental biology from very basic to the very advanced. So development basically describe the changes in an organism from its earliest beginnings throughout the maturity. That is what exactly we can call development. Don't get confused here. So friends, every one of you listen my statements very clearly. First, we have to discuss gamete formation. We have to discuss fertilization, cleavage, gastrulation, organogenesis, and finally growth. Now, what is this developmental biology is actually, we are going to read every stage. We are going to read every stage of development by using varieties of model organisms. What friends? We are going to discuss varieties of, developmental stages by using various type of a model organism. An example, we should not read the fertilization only in the mammals. We have to understand the fertilization in various organisms. An example, we have to read the fertilization in the sea urchin. We have to read the fertilization in the mammals. We have to read the fertilization in other organisms as well. So if you look into the cleavage, we have to understand the mechanism of cleavage in insect. We have to understand the cleavage in mammals. We have to understand the cleavage in amphibians. We have to understand the cleavage specifically in the sea urchin. So we are going to read different stages of the development by using different model organisms. These are said to be the developmental biology. So it's a very broad unit. And you have to understand. I can say that 80% of the concept to be understood, 20% of the concept to be remembered as well. All right. Now let's start the first stage, which we can call the gamete formation. So what we generally call it as gametogenesis, production of the gametes in the male as well as production of the gametes in the female also. So first we will discuss the production of the male gamete, which we call it as the spermatogenesis. It's not difficult, so easy to understand. Just focus on the lesson very correctly. Please turn off your audios and videos. Now, what is called as spermatogenesis? As the name itself indicating, genesis represents what, friends? Production. And sperma represents what, friends? Formation of the sperm. So the mechanism of the production of the sperm, we generally call it as the spermatogenesis. No point of getting confused. Understand all the time. The process by which diploid cells undergo meiosis, which is a reductional division, to form 4 haploid sperms. So every one of you understand, if a male ejaculating the sperm today, remember everyone, if a male ejaculating the sperm today, whatever the sperm that he is ejaculating today, actually that sperm produced, before 65 to 75 days. Now remember, the spermatogenesis or the production of the sperm takes about 65 to 75 days to complete. And remember my words very clearly without confusion. One ejaculation, okay, one ejaculation by a male, it generally contains 200 to 300 million spermatozoa daily on average. In simple words, one ejaculation contains 20 to 30 crores of the sperms. But out of that 20 to 30 crores of the sperm, hardly one sperm will go and fuse with the egg cell, which give rise to the zygote. Don't get confused. Now the question is very clear, where this sperm is getting produced. So the sperm is getting produced in the testes at a specific location. So now if you look into the cross section of the testes, you all people can see this picture very clearly here. There are many seminiferous tubules are there within the testes. So everyone remembers, seminiferous tubule is the region where the sperm production will be starting. Don't get confused here. Understand the concept correctly. So listen. Seminiferous tubules are located within the testes. Subsequent creation of gametes, namely spermatozoa. We will discuss one by one. The cell that is majorly required for the spermatogenesis is Leydig cells, Sertoli cells. So some cells will provide the nourishment, some cells are involved in the protection also. We'll discuss one by one. And major important, you have to remember that the hormones that are actually required for this particular spermatogenesis is GnRH. We already studied in the endocrine system. Gonadotropin releasing hormone is majorly controlling FSH and LH. And I believe in the endocrine system, I have explained you. FSH is generally involved in what? Spermatogenesis or the sperm formation. And what the LH is involved, LH is actually involved in the synthesis of a male sex hormone, which is involved in the second development of secondary sexual characteristics in the male, that is the testosterone. But these two are under the control of what? GnRH. Now every one of you focus from this particular slide and try to understand the concepts very clearly without having a confusion. Just understand. Spermatogenesis is a process by which diploid cells undergo the meiosis to form finally haploid sperms. Now friends, don't get confused. Just now what I told to all of you, I told you that sperms are actually produced in a region which we generally call it as the seminiferous tubule. I want every single student has to look into this beautiful picture. What is this actually? This is the membrane of the seminiferous tubule. Understand my words. This is called as a membrane of the seminiferous tubule. And what is this entire region we said it to be? This entire region we generally said it to be the lumen. This will be the membrane of the seminiferous tubule. This is the inner space or lumen of the seminiferous tubule. Now, every student understand my words very correctly. Look into this. Look into this. This membrane of the seminiferous tubule, we generally call it as what, friends? Basal lamina. Don't get confused over there. Now, every student try to understand my words very correctly. Now, near this particular basal lamina, everyone understand, there are certain cells are located, certain germ cells are located near this particular seminiferous tubule. So these are said to be the germ cells. Now, what happened to this particular germ cells, friends? These germ cells which are located near the basal lamina, as a part of the development, remember my words, as a part of the development, they are getting differentiated into spermatogonia. No point of getting confused, friends. Actually, it is a germ cell. As a part of the development, it is getting differentiated into the spermatogonia. Now, all the students understand. This spermatogonia undergo the meiosis 1, and it is going to give rise to the primary spermatocytes. Primary spermatocytes undergo the meiosis 2, and it is going to give rise to the secondary spermatocytes. Secondary spermatocytes giving differentiated into spermatids. Spermatids are finally differentiated into the mature spermatozoa, and finally, friends, they are getting released into the lumen. So once again, remember the process by which a diploid cell undergo the meiosis to form 4 haploid sperms. No point of getting confused. Sperms are produced generally from the primary spermatocyte. This is a primary spermatocyte. The primary spermatocytes undergo the differentiation, giving rise to the secondary spermatocytes. Look into this picture. No point of getting confused. You will get a very good idea. Understand, all of you. Look into this. Look into this. This is the primary or primordial germ cell. Where this particular primordial germ cell is present, you can see that it is present on the outer wall of the seminiferous tubule, such as the basal lamina. Now, this primordial germ cell, first, it undergoes various mitosis divisions. It undergoes various mitotic division, and finally, friends, it is getting differentiated into what? It is getting differentiated into the primary spermatocyte. Now, that primary spermatocyte undergo the differentiation. How it undergo the meiosis 1, giving rise to the spermatocyte. That is the secondary spermatocyte. Secondary spermatocyte further undergo the reductional division meiosis 2. It is going to give rise to the spermatids. So 1 is divided into 2. 2 is divided into 4 by the meiosis 1 and meiosis 2. And those spermatids can convert it into the mature sperm. How they are converting into the mature sperm, we're going to discuss now. Understand. Every one of you focus on the picture. Spermatocytogenesis. Remember, at the stage of the puberty, the germ cells, where these germ cells are located, outer wall of the seminiferous tubule. At puberty, the germ cell awaken, and they are going to start the actual process of the spermatogenesis. What we call that particular outer germ cells, we can call the spermatogonia. There are 2 types of spermatogonia. One is called as the type A spermatogonia, as well as the type B spermatogonia. Friends, this particular slide is a very important slide because you can expect the question in the form of a flow chart. So you have to remember the question compulsory for the exam. Look into this. This is called spermatogonia, which is the germ cell. This spermatogonia differentiate into type A spermatogonia. It is differentiated into type B spermatogonia. And this type A spermatogonia will be remain as it is. Whereas, friends, type B spermatogonia, can be differentiated further. Understand everyone. So every student remember, formation of this type A spermatogonia and type B spermatogonia. This process only we call it as a spermatogenesis. You have to understand that point compulsory for the exam. Now, friends, this type B spermatogonia, it undergo meiosis. It is going to give rise to the primary spermatocyte. Primary spermatocyte undergo further meiosis, giving rise to the secondary spermatocytes. In simple words, friends, formation of the spermatogonia, we can call spermatogenesis. Formation of the, what we can say that spermatocytes, we use that word. Remember everyone, spermatogenesis. I want you to remember the statements compulsory. Spermatogenesis is a process of the formation of the both primary and secondary spermatocytes. Now the primary and secondary spermatocytes, they get differentiated into spermatids. So you can see the picture, friends. Initially, the spermatids are completely round. As the time progresses, they converted into the elongated spermatids. So formation of the spermatids, we call it as spermiogenesis. Spermiogenesis. Then finally, friends, that elongated spermatids finally differentiated into sperm cell. So final that sperm cell, what do we call it as? We call it as the spermiation. No point of getting confused over there. And friends, what's the very interesting point here? The very interesting point, finally, once the sperm got synthesized, or look into that, the sperm is actually released where? They are actually released into the lumen of the seminiferous tubule. Don't get confused. Now, from the spermatogonia till the spermiogenesis, where they actually formed on the membrane of the seminiferous tubule. But once the sperm got released, the sperm is finally getting released into the lumen. So it is very important you people has to remember compulsory these terminologies, compulsory for the exam. Now, all of you please try to understand how the sperm look like, what is exact structural composition of the sperm, how the sperm is getting maturated. Is it really necessary for the sperm to become maturated? Without maturation, does the sperm associate with the egg cell or not? Everything is important here. All are important with respect to the statement related question. If you observe the structure of the sperm contain head. We will discuss that. And head further contains what? Head further contain nucleus. Head further contain acrosomal vesicle. You got that point. Then below the nucleus, it contains centrioles. Then below, you can say that centriole is like a neck. Okay. Below the neck, we have a mitochondria, not a one mitochondria, two mitochondria. The sperm having a multiple mitochondria. Then we can call the tail portion. If you observe the tail portion very clearly, friends, it contain flagellum as well as it contain tail sheath also. Don't get confused. It contain flagellum as well as it contain the tail sheath. This is a simple structure of a sperm. Now let's try to understand every part elaboratively. Now the first one, head. What I told you very clearly, head contain haploid nucleus and a propulsion system. This is a very unique statement. Now I want every one of you to answer. Shafia, tell me very clearly. Tell me, Swati, Ritika, Akila, what is the meaning of a propulsion system here? How the nucleus is getting moved? Every one of you answer. If something wants to move inside the nucleus, what actually it required? SSV, if something wants to move inside the nucleus, what exactly it requires? If an organelle wants to move inside, what they actually require? Everyone. So if a nucleus, very good, very, very good, Maria. If the nucleus wants to move inside, compulsory, what do they require? They require the cytoskeletal apparatus. Correct or not? Very good. They basically require an important protein here, dynein. Very important protein. So friends, that dynein is the one which is actually acting like a what protein? I hope every one of you remember. It is generally acting like a motor protein. And that motor protein is actually responsible for a movement of this particular propulsion system. The propulsion system is made up of dynein. We will see that. So it contains a nucleus and a propulsion system. And what else it contains? It also contains a sac of enzymes. It also contains a sac of enzyme that enable the nucleus to enter the egg. No point of getting confused over there. Very easy to understand. What is the meaning of a sac of enzymes? We all people commonly know that they basically contain acrosome vesicle. Every one of you know very well, acrosome vesicle contain abundant amount of hydrolytic enzymes which are actually fusing with the outer covering of the egg. But that is not what important here. What is very important here, how this acrosome vesicle is actually formed. That is important here. The acrosomal vesicle, we call it as the modified secretory vesicle. Very important statement to be remembered. They have actually derived from the Golgi apparatus. There is no point of getting confused over there. So together, acrosome and nucleus constitute the head of the sperm. No point of getting confused. Then friends, next one will be the neck. Very important, right? What I told you, neck basically contains what? Centrioles, which is actually giving rise to the axial filament of the neck. So remember, neck is basically made up of the centriole. And the most important, right? Sperm always moving continuously in the vaginal cavity. Sperm can withstand in the vaginal cavity up to 48 to 72 hours. And never think that a sperm will be remain restricted to one single place. Sperm will be moving all the time, every time. So if sperm wants to move continuously inside, it require abundant amount of the energy, which is a continuous source of energy. That's the reason the middle part of the sperm contain numerous mitochondria. It gives energy to the sperm to swim in the female genital tract. No point of getting confused. That is what middle piece. Now, look into this. Now I want every single student has to look into the picture correctly. What is this, friends? This will be the nucleus. And what is this? You can see that this is the acrosomal vesicle or modified secretory vesicle, which is actually made up of Golgi apparatus. Don't get confused. And this is what, friends? Neck, which is made up of what? Centrioles. And very important, next, middle piece contain what? Mitochondria. And what is the tail is made up of? Tail is absolutely made up of the flagellum. Now, I want you to remember a very important statement here. What is that important statement? Remember my words, until the sperm formation completed, listen everyone, you can see here the cytoplasm that is present in the sperm. It is present as a separate body. It is present as a separate region. This is generally we can call a casting body. What we can call casting body, or you can use the word hanging region. No point of getting confused here. So this residual or casting or hanging region of the sperm, we generally call it as the cytoplasm. Once the sperm formation is completed, friends, obviously this residual body will be casting off. It will be separated from the sperm. No point of getting confused. So remember, friends, what are the simple differences? Golgi apparatus is the one which forms the acrosomal vesicle. Centrioles are the one which is making a collar around the neck. Microtubules will form the flagellum. Mitochondria will provide the energy. Excess cytoplasm cast off as a residual body, or released as a residual body. So friends, this entire process will generally takes place 65 days. That is the reason if a man is ejecting the sperm today, that sperm generally produced before 65 days. No point of getting confused over there, and it is very easy to understand. Now, friends, what we discussed, mitochondria is providing the constant energy for movement. Now, the question is that who is helpful in the movement? Undoubtedly, friends, the tail portion of the sperm is exclusively responsible for this particular movement. No point of getting confused over there. Now, the question is super clear here. How? How this particular tail is involved in the movement? You know, friends, 99.99% of the sperms, they will always show whiplash movement, or you can call it whipping movement. So always remember, friends, sperm never ever move in a straight direction. Sperm move like a fish movement. Fish always move like this. They never move in a straight direction, which we generally call as whiplash movement. So most of the sperms also will show the mechanism of the whiplash movement only. Certain sperms, such as sperms of Ascaris, they will generally show, friends, amoeboid portion. Don't amoeboid motion type of a sperm movement. Don't get confused. So how does this particular tail is helpful in the movement, friends? The major portion of the flagella. This is very important, friends. So this is a tail portion, and this entire portion, we generally call it as the flagellum. And friends, remember, the suppose this is the flagellum. Now what I have done, I have taken the cross section of the flagellum. The major cross section of the flagellum, we generally call it as the axoneme. This is called axoneme, this entire region. Now, what is the major composition of the axoneme? Everyone understand, try to understand it. So look into that. The core of the axoneme consists of the two central microtubules. This is called one microtubule. This is called as the second microtubule. You can see that, which are actually present where? Which are actually present in the central portion. So the central portion of the axoneme contain the two central microtubule, which are surrounded by group of other microtubules. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 microtubules of a doublets are surrounding the two central microtubules. That's the reason we call it as the 9+2 arrangement. We call it as what? 9+2 arrangement. Don't get confused. And just now I told to all of you, if something wants to move inside, what does it compulsory required? It compulsory required this specific motor proteins. Very important, friends. So the motor protein that is associated with this particular, microtubules are dynein protein. So if the dynein protein got mutated, if the dynein protein got absent, friends, sperm cannot move at all. Now remember, here, this is the microtubule, right? If you observe the structure of the microtubule, it is actually doublet. This is called as A type of the microtubule. This is called as B type of the microtubule. No point of getting confused over there. And friends, here comes a very important point. Very, very important. In the center, there are two microtubules are there. On the outer region, there are 9 microtubules are there, which are doublet in nature. Now, question is, this is one doublet. This is another doublet. This is another doublet. Now, the question is super clear how all these doublets are moving. How all these doublets are connected with each other. That's an important point here. So one doublet can be connected with the other. Even the central microtubules are also associated with each other by a specific protein, which we can call the nexin protein. Don't get confused with that point. Very important to remember. And where all these 9 microtubules are projected, I mean to say which direction they are actually located. If you look into the direction, friends, remember all these particular 9 microtubules are projected towards the center. Now, how they are projected? Only towards the center with the help of a certain spoke-like structures, which we generally call it as the radial spokes. I want you to compulsory remember. So radial spokes are the one which are projecting this particular microtubules towards the central direction. So easy to understand this point. So I will repeat this completely. So if the sperm wants to move, the active part of the sperm that is involved in the motility is the tail. So tail is basically made up of a flagellum. And if you take the cross section of the flagellum majorly made up of the axoneme. And if you look into the structure of the axoneme contain two central microtubules which are surrounded by a 9 microtubules which are doublets, and all the 9 microtubules are projected radially towards the central 2 microtubules with the help of a radial spokes. And one doublet can be associated with the another doublet with the help of a nexin protein. And if this microtubules wants to move, they have to compulsory associated with a major motor protein that is said to be the dynein protein. This is a very simple structure of the axoneme of the flagellum of the tail. Don't get confused. Now, here comes a very important step where we people expect the questions in the exam. Now, finally, sperm got synthesized. Now, whenever the sperm got synthesized, friends, remember, we can use the word. Whenever sperm got synthesized, we can use the word, it got matured after the nourishment. We can use the word, it got matured after the nourishment. But we can't say that it got matured 100%. It's not matured 100%. What is the meaning of 100% maturation? 100% maturation is actually, representing the sperm acquires the ability to fuse with the egg cell. Whenever sperm acquires the ability to fuse with the egg, then only, friends, we can say that the sperm got completely matured or 100% matured. Now, question is very clear where this 100% maturation of the sperm is actually taking place. That is an important point here. The 100% of the maturation of the sperm actually taking place only in the female genital tract. Remember, not in the male genital tract. Whenever sperm enter into the female genital tract through the vaginal cavity, especially in the fallopian tube, there only the sperm is undergoing the maturation 100%. So the process of maturation of the sperm 100%, that is what exactly, friends, we call it as a capacitation. Capacitation is the mechanism of 100% of the maturation of the sperm. Don't get confused. And it only happens in the female genital tract. So listen, everyone, the final stage of maturation doesn't happen. The final stage of maturation doesn't happen unless they are in the reproductive tract. No point of getting confused. Sperms cannot fertilize the oocytes when they are newly ejaculated. Such an important statement to be remembered. Sperms cannot fertilize the oocytes when they are newly ejaculated because they did not undergo the complete maturation process. So when I use the word capacitation or a final stage of the maturation, the question is very clear. What is the meaning of that word? What is the meaning of the word called as capacitation? Which means, the sperm undergoes various physiological changes. That's all. I'll tell you what exactly those changes. The sperm undergo various physiological changes. After the physiological changes only, the sperm acquire the ability to fuse with the egg. That is called capacitation. And what is that? Where that process is happening? That process is only happening on the female genital tract. Now you can answer a question, what is the reason? What is the reason why it has to happen only in the female genital tract? Suppose this is the sperm head. Every single student try to understand. On the head of the sperm, certain cholesterol molecules are present. Even on the head of the sperm, certain albumins are also present. Now, what is our ultimate goal? In order to make the sperm to become completely matured, we have to remove that albumin. We have to remove that cholesterol. So removing of this albumin and cholesterol from the head of the sperm, that is only we people generally call it, friends, physiological changes. No point of getting confused. And how can we remove the cholesterol? How can we remove the albumin? We require certain chemical substances in order to remove this. And where these chemical substances are generally found, these chemical substances are generally found in the female genital tract only. That is the reason only we can say that the process of capacitation exclusively taking place in the what we can say female genital tract only. So friends, very important. How can you differentiate or how can you confirm? That's also very good point. How can you confirm whether the capacitation occur or not? If the process of capacitation did not occur, the acrosomal reaction never ever takes place. What is the acrosomal reaction? The hydrolytic enzymes will be released from the acrosome and they will start digesting the outer layer of the egg cell. But that reaction, if it is not taking place, which indicates that the process of the capacitation is not completed. That is the difference that we can compare whether the sperm undergone the capacitation or not. Now, no point of getting confused. This process of capacitation basically require a process of a simple signaling. Very simple. Because here we are expecting the questions in the form of a statements only, friends. So I encourage everyone to remember the statements very accurately. Listen, capacitation actually involves the destabilization of the sperm head membrane, allowing greater binding between sperm and oocyte. So such an important statement here. In single word, what we are using the word, we are destabilizing the sperm head membrane. This is our ultimate goal. Now the question is here, friends, how? How can we destabilize the sperm membrane? That is an important question. All of you please look into this. This is the membrane of the sperm head. So you can see that on the membrane of the sperm head, there is a presence of cholesterol. There's a presence of albumin. What should I do now? I have to remove these two. That's all. That is my ultimate goal. Once I remove these two, the sperm head which contain acrosomal vesicle, it becomes super active. But friends, if we want to remove this, there is a involvement of the signaling. Now, so every student understand. When I remove albumin and cholesterol, you know what happened, friends? The sperm membrane also become more negative. Now, question is very clear. Why the sperm membrane becomes more negative? What is the reason? Because on the sperm membrane, that is also a question, friends. On the sperm membrane, more amount of the potassium efflux channels are present. What kind of channels are present? More amount of the potassium efflux channels are present. So friends, since more amount of the potassium efflux channels are present, more potassium comes out of the cell. Whenever the more potassium comes out of the cell, you look into the picture, what are coming inside? Bicarbonate is coming inside. You can see the picture. Calcium is coming inside. That is very clear. Now, friends, whenever the bicarbonate and calcium comes into the inside, what happens? Calcium and bicarbonate going to activate an important effector molecule. Don't get confused, which is called as the adenylyl cyclase. You got the point. Now, friends, what is this adenylyl cyclase is going to do? You know very well. Adenylyl cyclase convert the ATP finally into the cyclic AMP. I believe every one of you also remember very clearly What is the function of the cyclic AMP? Cyclic AMP is the one which is going to activate a protein kinase A. Now, friends, this protein kinase A is actually doing two functions. Such an important statement it is. I want every one of you has to remember this important statement. Protein kinase A is actually doing two functions. The first function, see, you tell me what is the function of a kinase. We all know very well the function of the kinase is the phosphorylation. So it is phosphorylating two proteins. It is phosphorylating PTK protein also. It is phosphorylating PTP protein also. But friends, whenever it phosphorylate the PTP protein, protein tyrosine phosphatase, it inhibits. Whenever pKa add the phosphate group to the PTK, PTK will become active. So friends, whenever the PTK will become active, it stimulate the process of the capacitation. So point is very clear. If the process of the capacitation wants to occur, it is very important, friends. The PTK should be active. If the PTK wants to be active, compulsory there should be activation of pKa, which is activated by cyclic AMP. Cyclic AMP synthesized by the adenylyl cyclase, and adenylyl cyclase going to activated by the bicarbonate and calcium. Bicarbonate and calcium is entering inside by the efflux of the potassium. Potassium is going out upon removal of this particular albumin and cholesterol. This is a simple signaling process that is involved in the process of the capacitation. No point of getting confused over there. So every one of you very quickly answer. Don't take too much of time. Since it's a first day, I'm going little slow, okay? From tomorrow onwards, I want every one of you to be more active. I'll go little speed, okay? Because revision course, we have to do little speed. Ritika, I'm telling you, I'll send you to the 11th standard. As the day progresses, you're always forgetting the things. Just now I told you, right? Capacitation only occurs at a specific area. Correct, na? So actually, the sperm enters via vaginal cavity. But the question is what? The question is not about how the sperm is entering. The question is about where it is getting complete matured. So where is that specific region? That specific region, we generally call it as what? Fallopian tube. No point of getting confused over there. Okay. Try to understand it correctly. Right. Good, good. No problem, no problem. Okay. Now, spermatogenesis is induced by which hormone? So, of course, spermatogenesis is induced by the follicle stimulating hormone, which is under the control of the GnRH, then followed by the hypothalamus. No point of getting confused over there. Now, no point of confusing. Think properly and give the answer. Correct sequence of correct sequence of cell states in spermatogenesis is. Correct sequence of cell states in spermatogenesis is. Slowly, slowly. Don't do so fast. So first, spermatogonia. Spermatogonia is what I told you, germ cells. They are differentiated into spermatocytes. Correct, na? Spermatocytes are primary spermatocyte, secondary spermatocyte. Secondary spermatocytes differentiate into spermatids. Spermatids differentiated into the spermatozoa. Correct, na? Correct answer will be the D. Don't get confused. Correct, correct, correct. Don't get confused. Okay. Now next, slowly, slowly think and answer. How many secondary spermatogonium are required to make 400 spermatocytes? Selvi, what is the answer for this? Bhavani, everyone answer this question. Sneha, Yashoda, what is the answer for this? How many secondary spermatogonium are required to make 400 spermatocytes? I have finally told to all of you friends, one spermatogonium is going to give rise to the 4 haploid sperms. One spermatogonium is going to give rise to the 4 haploid sperms. So here they have asked us for the 400 spermatocytes. So basically 100 are generally involved because very good. They undergo the meiosis 2, which is the reductional division. So we get total of 400 when 100 secondary spermatogoniums used. Don't get confused. Good. Again, think and answer the question correctly. No point of confusion. I'll read the question. The question says that in an experiment, sperm got removed from the epididymis of a male mouse, was added in a dish containing appropriate media and oocyte. Hence, friends, no fertilization was observed. However, when sperm from the epididymis were directly placed in the uterus of an ovulated female, she became pregnant. What these observations are suggesting that the question says that we have removed a sperm from the male mouse and we have added this sperm to a petri dish which contained the egg cell. But what happened? No fertilization was observed. Second condition, what we have done, we have taken the sperm, we have directly kept in the uterus of a ovulated female. She became pregnant. What is the reason they are asking? Okay, what is the reason they're asking? Now, if every one of you look into the option 1, it says that the sperm needs to travel some distance to attain the fertilizing ability. As all of you know very well, friends, even though sperm travels for a very longer distances, friends, it is not going to attain the ability to undergo the fertilization. No. Option 2 says that the oocyte secretes some biochemicals or the factors which help the sperm to fertilize. No, that is wrong. The hormones in the body, no. Hormones are not at all involved in the, fusing process. Fusing process is completely acrosomal vesicle reaction. Option 4 says that the content of female reproductive tract interact with the sperm activated for the fertilization process. Correct, na? Hence, the correct answer will be the number 4. Okay. Everyone quickly confirm. See, this is an introductory class. Now, this class I am telling you, especially to the previous batch people, I will focus from the cleavage, mostly questions. I will solve here itself 2023, 2022 questions. You people make sure that you will revise the topic one more time and you will answer every single question that I will show you in the class, especially cleavage, gastrulation and organogenesis. So attend every class regularly. Clear everyone so far? Quickly confirm. It's a simple one only. All right. Okay, kindly work very hard for this 50 days. Okay, kindly don't give up in the middle. When we started something, first few days, we will be very much excited. So that excitement will stop. Not this month, SSV. Okay, mostly 15 days, dear. That's all. Mostly 15 days. Okay, or it will go 16 days. That's all. I'll complete it everything. Not that many days. Okay. In middle 4 days, you are having a paper 1. After that, I will finish it very faster. Okay. Not the whole month. Okay. Now, everyone, please join at sharp 11:30. Sharp 11:30. We are going to start the revision. Okay. Of the unit 6. Everyone join compulsory. Okay. In the 2:30, we will start the chapter of unit 11, evolution. Okay. Now I'll do the plant physiology revision so fast. Okay. Very fast. At the end only, I'll solve the questions. All right. 99%, I'll take from the app. In case if it is batch was not active, I'll share the Zoom link. Okay, I'll share the Zoom link. All right, everyone join at sharp 11:30. Take some rest and start your preparation again. Always think that today is the last day of your preparation and give your best in your preparation. All right. Okay. Thank you all. Thank you.